Question 4. Leptin is a protein hormone with a role in the control of appetite in humans. The leptin gene is located on chromosome 17. So this comes back to our understanding that some, proteins can do a lot of different things. They can be hormones, like this one, like insulin. They can be structural proteins like collagen. They can be antibodies, they can be enzymes, but all proteins are made from a gene. Yeah? Okay, so state what is meant by the term gene. Okay, so key points about genes is that they're a sequence of bases in DNA that code for a specific protein. Sequence of bases in DNA that code for a specific protein. Yeah, I think the sequence of bases should be one mark and code for a specific protein should be another mark. Could I flesh that out to cover my bases a little bit more? Sequence of bases in DNA that code for the synthesis of a specific protein. I think that should be enough. Let's see. Describe the role of tRNA in the production of leptin. So describe the role of tRNA. Okay, so remember, tRNAs are involved in the process of translation. It's 3 marks. So what do tRNAs do? Yeah, okay, so tRNAs, they bring a specific amino acid. tRNAs bring specific amino acids to a ribosome during translation. So where possible, what I'm trying to do is just kind of put a lot of information into each of my sentences as much as I can. So bring specific amino acids to a ribosome during translation. So what do they do though? They bind specific So tRNAs bind specific amino acids. They bring specific amino acids to a ribosome during translation. Okay. Their anticodon binds to codons on the mRNA. Bind a specific amino acid, bring specific amino acids to a ribosome during translation. The anticodon binds to codons on the mRNA and allow. So it doesn't say describe the role of a tRNA, it says describe the role of tRNA. So it could be many. And allows amino acids to be joined in a sequence determined by the sequence of codons on the mRNA. Okay, the anticodon binds to codons on the mRNA by a complementary base pairing. Okay, and allows amino acids to be joined in a sequence determined by the sequence of codon on the mRNA. I mean, should I say something about the genetic code? I'm not sure. Okay, I think that should be it. Yeah. Describe the role of tRNA production of leptin. Allows amino acids to be joined in a sequence determined by the sequence of codon on the leptin mRNA. Could I say which ensures the correct primary structure? Of leptin. Okay. Yeah, that was a bit of hard work to flesh out. Okay, describe how the primary structure of leptin enables it to be soluble in water. Describe how the primary structure of leptin enables it to be soluble in water. So it's a describe question, it's 3 marks, but when I see soluble in water, where I go to is globular proteins. Okay, and in order to be a globular protein, you need to have your hydrophilic R groups on the outside and the hydrophobic R groups on the inside at the core of the protein, and the hydrophilic R groups on the outside of the protein interacting with water. Okay, so it's an awkward question because that's more to do with the tertiary structure. Right. Describe how the primary structure of leptin enables it to be soluble in water. Okay. Don't want to overthink it. We're still early in the paper, so just got to get on with it. I know there's going to be stuff about hydrophobic amino acids and hydrophilic amino acids, R groups. Okay, maybe I'll just begin by defining the primary structure. So, primary structure is the sequence of amino acids in the polypeptide chain. Okay. Leptin primary structure. Leptin primary structure must contain amino acids must contain hydrophobic R groups and hydrophilic R groups. So the leptin primary structure must contain both hydrophobic R groups and hydrophilic R groups. Must contain amino acids with, I guess. Must contain amino acids with hydrophobic R groups and amino acids with hydrophilic R groups. Okay, so as a result of R group interactions, a tertiary structure is formed. A tertiary structure is formed that has or maybe I want to say that as a result of the primary structure. So as a result of the R group interactions determined by the primary structure, okay, a tertiary structure is formed that has hydrophilic R groups on the surface of the protein, okay, on the surface, so it can interact with water, and hydrophobic R groups in the center of the protein, in the center or core of the protein. Okay, it does say soluble in water. Maybe I can be a bit more efficient with how I'm getting to the point here, but hydrophobic R groups in the center core of the protein, away from water. On the surface, okay, now soluble in water, yeah, surface, hydrophobic R groups interacting with water. I just wonder if, because they've said soluble in water, whether they're expecting a description of what's going on there. Anyway, right, B, several mutations of the leptin gene have been identified. All these mutations are frame shift mutations that result in shortened primary structures. A frame shift mutation involves the insertion or removal of one or two nucleotides from a gene. Describe how a frame shift mutation could result in the production of leptin with a variety of shorter primary structures. So describe how a frame shift mutation could result in the production of leptin with a variety of shorter primary structures. Okay, so frame shift mutation alters the reading frame of the gene after the mutation site, let's call it. Okay, so it alters the reading frame. So this changes the codons. This changes every codon after the mutation. Okay, and what could happen as a result of that is we could introduce, so this could introduce premature stop codons. So because we've changed the reading frame, some of the new codons, in inverted commas, could be stop codons. Yeah. So this would stop translation earlier, and produce polypeptides with fewer amino acids. And that's question 4. Okay, I mean, it's 2 marks. Yeah, so frame shift, so we say what the frame shift mutation is, we say what the impact is, changes every codon after the mutation. This introduces premature stop codons. This would stop translation earlier and produce polypeptides with fewer amino acids.